B&H production presents how we code for a specific structure or protein. Yeah. Right. We're basically going to be talking about E. coli and the fact that it has genes which code for two specific enzymes called beta-galactoside permease, also known as lactose permease. And this enzyme allows lactose uptake, so it allows the E. coli to take in lactose. And the other enzyme is beta-galactoside, if I pronounce that right, also known as lactase, which allows the enzyme E. coli bacterium to hydrolyze lactose to galactose and glucose. Yep, so you can respire with that. We're just going to talk about something quickly. If the bacterium is grown on a medium containing glucose, these enzymes are not produced. And if the genes are basically switched off. If it is placed in a medium containing only lactose, then the genes are switched on and these enzymes are produced. Now, what determines this is something called the Lac Operon. It's the length of DNA made from structural genes and control sites which code for specific proteins and other bases which determine whether or not the gene will be switched on. So basically, depending on your environment, it determines if you need something or not, basically. And going into detail with it, this is basically what it looks like, if we can create a model of it. As you can see, we've got the regulator gene, the promoter gene, operator gene, the gene which codes for beta-galactoside gene, which is also known as lactase, and the other bit, beta-galactoside permease, which is also known as lactose permease, that bit there. Yeah. Brilliant pointing by Powledge. So the theory of it is that the RNA polymerase, you must have heard of that enzyme before, it basically allows nucleotides to be joined together and form RNA, binds to the length of DNA called promoter RNA, so that bit there. So the RNA polymerase binds to the promoter, and RNA polymerase must bind in order to begin the catalysis of the transcription process of mRNA from structural genes. So basically what it's saying is RNA polymerase must bind to it in order for these two here to be coded for. So the two enzymes to be coded for. RNA must bind here. And so the next step includes the operator. It's right next to the promoter. Promoter and the operator. Yeah? If nothing is bound to the operator, then the promoter is available for the RNA polymerase to bind to.
Thank God for that.
Hence the structural genes can be expressed. Expressed basically means the structural genes can be actually coded for. So these two can be coded for. Yeah? And however, another stretch of DNA on the operon called the regulator codes for a protein called the repressor protein. The repressor protein has two active sites. One of these fits the operator DNA and so binds with it. When this happens, the promoter is blocked. So basically, something comes along, right? Basically, and it binds to the operator here. This creates a gene. This creates a protein which binds to the operator, stopping it from working. When this happens, the promoter is blocked. When the protein this creates binds to this gets blocked. So the regulator protein, also the proper word for it is the repressor protein, which is coded for by the regulator, binds to the inactive site on the operator and blocks the promoter. So now RNA polymerase can't bind to the promoter, and so the two enzymes can't be coded for.
Exactly.
However, the repressor protein also binds with lactose. When this happens, the shape of the repressor protein changes and it does not fit the operator DNA. So we have, if you grab a pen, that's it, for a pen, yep. So we have little molecule of lactose here, right?
Heart.
Sorry, it's a heart. I'm left-handed. We have a molecule of lactose here. It comes along, and it binds to the repressor protein on the operator.
Sure.
So now the operator is freed. So the repressor protein moves out the operator out here, and now the promoter's active site is open again. So RNA polymerase can happily bind to it, and the RNA polymerase. So that's our lactose binded with our repressor protein, and so the operator is free, and now the promoter can bind to it, and RNA polymerase can bind to the promoter, and these two enzymes can be coded for.
And that's a reaction back.
Yeah,
To its original form.
and that's how we get
Down.
beta galactoside and beta galactosidase. Right. Another brilliant production by Boris.